safety precautions to reduce a laboratory and risk exposure to potentially infectious microbes and limit the contamination and containment of the work environment and ultimately the community. According to the WHO, biosafety is a strategic integrated approach to analyzing and managing the relevant risk to human, animal, and other species and associated risk for environment. Three different topics, important topics and concepts in the field of biosafety exist: biological hazards, biocontainments, and bioprotections. The biological hazards mean that the potential risk of uncontrolled exposure to the biological agents with infectious capacity and harming the individuals. Biocontainments mean measures we use to prevent the infectious disease from leaking from research centers or other places where they may be produced, such as we see in coronaviruses. And bioprotection means measures taken to reduce the risk of loss, death, misuse, or intentional release of pathogens. We have some definitions like biosafety level in laboratory. Biosafety level or pathogen protection level is set up by containment precautions required to isolate dangerous biological agents in enclosed laboratory facility. We have 4 biosafety levels, and they named and ranged from BSL-1 to BSL-4. Each level has specific controls for containment of microbes and biological agents. The primary risks that determine the level of containment are infection. How many people come in the middle of the class? چند نفر قراره بیان رو؟ در ببرم. Each level has specific controls for containment of microbes and biological agents, and the primary risks that determine the level of containment are infectivity, severity of disease, transmissibility, and the nature of the work we conducted. Also, the origin of the microbe and the route of exposure and transmission is very important for us. As I said in previous slides, we have 4 different BSL, biosafety level. And from the bottom of triangle to the highest of it, the risk is increased. In BSL-1, we have low-risk agents that are not known to cause severe human disease, such as E. coli or Escherichia coli. In BSL-2, infectious agents of moderate risk with ingestion or mucous membrane transmission, such as influenza, Lyme disease, and Salmonella. In BSL-3, we have infectious aerosol transmission that may cause serious or lethal infection, but we have treatment for this disease, such as coronavirus, mycobacterium, tuberculosis, and Yersinia. And in the highest level, in BSL-4, the infectious aerosol transmission that may cause serious or lethal infection, and we don't have any treatment for them, like Ebola and variola virus. But what is biosafety in our laboratory? The biosafety for us is the lab coat is highly garment of your job. Wearing a lab coat is mandatory from the time of entering the laboratory to the completion of the work. And please enter the laboratory in a clean coat, and please close the buttons of the coat. The skin is a way to transferring the pollution and microbes to the body. The use of gloves during experiment is an integral part of your work in the laboratory. Please use disposable gloves that are placed in your work table in every session. The laboratory environment is completely contaminated due to the working with all kinds of viral, microbial, and antigenic samples. Therefore, such a place is not a good place for eat, drink, or smoke. Avoid consuming any food or drink in the laboratory. The possibility of contamination of your personal belongings in the laboratory is easily available. Do not put your mobile phone, glasses, bags, laptops, and any additional equipment on your desk, please. And finally, your work desk is in the laboratory. From the beginning to the end of the work is your workplace. Please keep your workplace tidy and your work surface clean during the test. At the end of the work, be sure to put the used gloves and cotton in the trash and organize your desk. Okay, this is biosafety, and we entering to the immunoassays that we today want to experiment them. What is immunoassay? Immunoassay is a biological assay for measuring the presence or concentration of a macromolecule in a small molecule, thought an antibody or antigen. But when we talk about immunoassays, we should know about two definitions: analyte and signal. Analytes is that the molecule that were detected or qualified or quantified by immunoassay, and signal is something that we can understood by our sense. So we have an analyte or antigen. When antibody bind to this analyte, they transfer some signals we can sense with our sense, can understood by our senses. Analyte, as I say, after binding the analyte to antibody, we have different type of signals. Some signals is visible, some color, some light, fluorescence, radiometry, turbidimetry, and light scattering. And based on the signal we sense, different type of immunoassays are classified. For example, precipitation, agglutination, radioimmunoassay, enzyme immunoassay, fluorescence immunoassay, and chemiluminescence immunoassay. Today we want to talk about these two first sections, the precipitation immunoassay and agglutination immunoassay. And others can be defined in other classes. When we talk about precipitation and agglutination, you should know there is some similarity and differences between these two experiments. In the precipitation and agglutination, both of them, when antibody crossing antigen, they perform and produce complex that we can see by our eyes. But the difference is the physical form of antigen. The physical form of antigen in precipitation is soluble means our antigens is soluble. But in agglutination, our antigens in particle form. Firstly, we want to talk about the precipitation. In precipitation, we have 3 different types of experiments. Some of them are qualitative, some of them is semi-quantitative, and some are quantitative. And based on the base of the work, we have 2 types, liquid or semi-liquid. And in the resume, we want to talk more about these experiments. The important test for you is qualitative immunoprecipitation that divided to 3 experiments: double immune diffusion, counter-immunoelectrophoresis, and immunoelectrophoresis, and we want to talk about the basis of these experiments. In double immune diffusion, you have a slide, and under the slide we have gel. We can punch two sides of the gel for loading the antigen and antibody in each side, in different sides. For example, here we load the antigen and here we load the antibody and let them to diffuse to each other. When they arrive to each other, a line can be seen that we named precipitate, and means that the complex of antibody and antigen is performed. The countercurrent immunoelectrophoresis is the same mechanism that previous experiment, but there is a difference, and here we use the electricity to increase the speed of diffusion, so we have the result by electricity. And like the previous slide, we can see the precipitation in the middle of the gel. But immunoelectrophoresis is an important experiment in the field of immunology. Anyone can describe this slide, can talk about this slide? What this slide wants to say to us? Please.
Seems that first the microbe, when detected by the naive B cell, becomes an activated B cell,
Yeah.
and this leads to increase in its numbers. And then after that, the B cells they differentiate. So some become they secrete antibodies, and some express immunoglobulin G,
Yeah.
and some are memory B cells, and some they're called high affinity immunoglobulin expressing B cell, but I don't know what that
Yeah,
is.
of course, that's correct. After the binding of microbe to an B cell, these B cells activated in the presence of helper T cell stimulation, and they presence and experience clonal expansion, and from this level they differentiate and have different fates. Some of them are experiences class switching, some of them experiences somatic hypermutation, and then it's lead to production of B cells that produce high affinity antibodies. Immune reaction is important for us in immunoassays. But what is the immune reaction? The immune reaction is tied to antigen-antibody reaction. This reaction is highly specific, and antigen binding site of the antibody could perfectly match to the antigen, like what we see about key and hole. But what is the epitope? An epitope, also known as antigenic determinant, is the part of antigen that is recognized by immune system in our world is antibody. So where antibody is bind is epitope under the surface of antigen. We have two types of antibody in our body, but mainly our body produces polyclonal antibodies. We say we have two types of antibodies: polyclonal antibodies and monoclonal antibodies. The polyclonal antibodies derive from different B cell clones, and each of these B cells produce antibodies that recognize a different epitope on the same antigen. But monoclonal antibodies only derive from a single B cell clone and only recognize a single epitope per antigen. We have different immune-based disease. Some of them are classified in gammopathies. We have two types of gammopathies based on the type of antibody: polyclonal gammopathies and monoclonal gammopathies. Polyclonal gammopathies is heterogeneous increases in immunoglobulins involve more than one cell line, as I said in previous slide. This type of gammopathy caused by any variety of inflammatory, infectious, or neoplastic disorders. But in monoclonal gammopathy, a single B cell is proliferate more than what we want, what we see in our body. So we have homogeneous increases in the gamma region of serum protein electrophoresis. We see this gammopathy in multiple myeloma and also in Waldenström macroglobulinemia. But what is the immunoelectrophoresis? As I say, in double immune diffusion, we have a punch in our gel, and we load antigen in here, in this circle punch, and we conduct the electricity to electropoietic to our antigen, electropoietic to separate its component by charge. And then the throws are then cut into the agar gel parallel to the direction of the electric field, and antiserum is added to the trans. Antibody and antigen then diffuse together toward each other and produce lines of precipitation when they meet the appreciate proportion. As you see, after antibody moderation during the gel, we can see like these diffusions in our gel. This is the normal immunoelectrophoresis gel of human serum, and you can use from this slide for comparing with the disease that related to the gammopathies. This is the real type of slides of human serum immunoelectrophoresis, and this is the slides we give to you to report for us the monoclonality or polyclonality. If you show in your slides more than one arc, this is one arc and this is more than one arc. if you see more than one arc in your slide, you should report it as polyclonal antibody, and if you see only one arc in your slide, you should report as monoclonal antibody. Okay, this is the first session. We have another 40 minutes, I think. The neutralization is the next experiment we want to talk about. It neutralization. Antibodies have different functions after binding to the antigen. Among them, we can mention neutralization, complement activation, opsonization, and also ADCC. After binding the antibody to the surface of an antigen, that it can be virus, bacteria, or other type of microbes, this antibody can lead to the neutralizing the biological activity of microbe. This function is mediated by a variable region of antibody, and it's only the first function of variable regions of antibody. Other mechanisms like complement lysis, opsonization, and ADCC is mediated by constant region of antibody. After binding of antibody to the surface of infected cell or virus or bacteria, the Fc region of antibody is present, and immune cells can bind to these antibodies by Fc gamma receptors. And these receptors mediated the activation of immune cells and lead to the lysis of bacteria, virus, or infected cell. But what is neutralization? Neutralization, we have two types of antibodies and differ from each other. Neutralizing antibody neutralizes and suppresses the biological effect of any antigens. But binding antibodies flag antigen for immune cells function. As I say, the Fc region of antibody leads to activation of immune cells such as NK cell, macrophage, and other immune cells. In this test, neutralization or ASO test, we want to talk about the toxin produced from Streptococcus. This is the same microscopy of Streptococcus, and group A Streptococcus in human can lead to pharyngitis and tonsillitis, and also we have pyoderma and skin infections during this infection. The pharyngitis and tonsillitis ultimately can lead to acute glomerulonephritis and acute rheumatic fever, and skin infections can lead to acute glomerulonephritis. But what is the challenge? Why the immune reaction against an other organism and microbe, bacteria, can lead to attack our immune cells, attack to our own cells? Why this happens? Please.
Because streptococcal exoantigens are similar to our antigens sometimes.
Yeah, of course. Sometimes the antigen that produced or expressed by an organism is very similar to our own antigens, our own proteins, and antibody produced against the antigen can bind to our own cells. For example, about Streptococcus, protein M is very similar to myosin heavy chain. Group A cell wall glycoprotein is very similar to glomerular basement membrane, and pyruvate is very similar to synovium. So, any antibody produced against these proteins can bind to our own cells and lead to the activation and inflammation and lead to the disease. The common diagnostic methods for streptococcal infections is culture by patient samples, ASO titration that we today doing this experiment, and anti-DNA therapy. But what is the ASO principles? In this test, we have an enzyme that we want to neutralize its function. This enzyme is streptolysine O, or SO. We name streptolysine O that produced from Streptococcus, SO and SLO. This enzyme is RBC hemolysis and also oxygen sensitive. But about the basis of this experiment, we can talk by this slide. If you have a serum and the patient is not produce antibody against the Streptococcus SLO, after adding the SO to this serum, due to the not presence of antibody, the SLO is activated and can release the RBC. So if you see the RBC, your patient serum don't have any antibody against SLO. But if you have antibody in patient serum, after adding the SO or SLO or sterilizing, or they all same. After adding the SO to this mixture, antibody can bind to the SO and inactivate this enzyme or toxin, and this is lead to the we don't see any lysis in our RBCs. Okay, this is the procedure of your work. You can take a picture from this. You should add these amounts to the tubes, and you have 5 minutes of incubation, and then we change your racks with the other racks that your result is ready for reading. Okay, as you see in previous slide, we have 2 controls for our ASFS. The negative control that only have RBC and buffer to ensure our RBC and buffer quality, and the positive control only have RBC and SLO to ensure our SLO quality that can release the RBCs. The serum dilution you use in this study is 1 to 100, and for example in number 2, serum is diluted to 0.6. And the final dilution is 0.006. Tab unit is needed to report it by you, and this unit is the last tube. You don't see any hemolysis in it. I should back and explain to you. After adding these amounts by this replica, you should read your tubes, and the last tube you don't see hemolysis. You should report as tab. For example, if the tube number 3 is the last tube you don't see hemolysis, you should add its unit as a 250 for tab unit. It's okay? The tab unit calculation is like this. We can take a picture from it again. Okay, one time again, we want to repeat the results. If you see lysis in your tubes, you don't have antibody in the serum, and if you have if you don't see any lysis in your tubes, you have antibody in serum and your sample is patient. But the tab unit upper than 250 is recognized as patient, and lower than it is healthy. Thank you. We have another session. If you want, we have to quick 5 minutes.
Yeah, sure.
Abbreviation test. As we say, be quiet. As we say in the previous session, we have different type of reactions: precipitation and accumulation. In previous session, 2 type of the reactions that we named flagellation, and the antigen in this test and procedure is in colloid form. In agglutination reactions, we have also 3 different type of reactions. The active agglutination is related to antigens that are found naturally on a particle. The antigen is on its particle, its own particle. And in indirect agglutination, we use from anti-human globulins to see the agglutination, and the passive agglutination is related to the particles coated with soluble antigen. We have a soluble antigen, we coat it on the surface of some particles like latex. Okay, if you want to talk about right test, we should know something about the brucella. Brucella is a zoonosis caused by ingestion of unpasteurized milk from infected animals or close contact with their secretions. It is also known as undeveloped fever, mild fever, and Mediterranean fever. We have some symptoms after infection of brucella, such as fever, chills, loss of appetite, sweats, weakness, fatigue, joint and muscle, and back pain and headache. And we have different type of diagnosis tests for brucella and divided into two types. Type 1 is non-serological test like blood culture, stool culture, urine culture, and CSF culture, and the serological tests that we today want to talk about them is Rose-Bengal test, Wright test, Coombs-Wright test, and 20 Wright test. Okay, the Wright test basis is active agglutination, and for doing this test, we firstly should do the slide test and then we go to the tube test. And I'll explain more about them in the next slides. Okay, the slide test for Wright, named Rose-Bengal test, is a rapid slide agglutination procedure. For doing this experiment, we should use 1 drop of patient serums and 1 drop of Rose-Bengal antigen, and we should mix them with plastic applicator, and then if we see the agglutination, we can say our patient serum is positive for this antigen. Okay, if the right slide test, as I said previously, the right Rose-Bengal test is positive for a patient serum, we should done the tube test to determine the exact titer of the specific antibody against brucella in patient serum. This is the procedure of Wright test. You can take picture. You should perform serial dilution by this method. You should add physiological serum by amount of 0.9 to tube 1 and add 0.5 to other tubes firstly. Then you add 0.1 of patient serum to tube 1 and transfer 0.5 from tube 1 to tube 2 and repeat this process in other tubes. That means after adding the 0.5 from tube 1 to tube 2, you should add 0.5 from tube 2 to tube 3, 0.5 from tube 3 to tube 4, and like this for tube 5 and tube 6, and in the tube 6 you should spill over 0.5 from tube 6. And then you add antigen and you should report the dilution of the serum that you see the agglutination. The last dilution, you see the agglutination. As you see in here, in this slide, the last tube that we see the agglutination is the dilution of 1 per 106 C. Based on the WHO in Iran, titer equal or more than 160 are considered clinically positive. After this test, after the Wright test, Wright tube test, we can perform 2-ME Wright to determine the exact type of antibody that leads to the agglutination in our Wright test. For this experiment, we can add the 2-ME, 2-mercaptoethanol, to our solutions, and this can be degrade the IgM, but it cannot affect the IgG. If you see the reputation of your result in the 2-ME Wright, the antibody of patient serum in the patient serum is in class of IgG, and IgM is not affect our results. The next experiment is related to latex agglutination tests we named CRP and RF. It is important to know in the first of this session, I say to you we have three types of agglutination reactions: active, indirect, and passive agglutination. But we have one other type, and you can see in this slide. As we say, passive agglutination is related to the test that our antigen is soluble and we bind them to carrier molecule to detect it by antibody in the serum of patient. But in some cases, we don't attach the antigen to the carrier molecule. We attach the antibody to the carrier, and we name them reverse passive agglutination. Okay? That's okay? Any problem? Okay. C-reactive protein or CRP test, we want to talk about it. CRP rise in response to inflammation. It is an acute phase protein of hepatic origin that increases following the production of interleukin 6 and its secretion by macrophage and T cells. Acute phase proteins are a class of proteins whose concentration in blood plasma either increase or decrease in response to inflammation. In healthy adults, the normal concentration of CRP varies between 0.8 to 3 mg per liter, and in different circumstances of inflammation, its amount is rise, like mild inflammation, active inflammation, severe bacterial inflammation, cancer, heart attacks, and rheumatoid arthritis, and their amount is vary, so you can determine the type of inflammation based on the amount of the CRP. We have different methods to detection of CRP like ELISA, immunoturbidimetry, nephrometry, rapid immune diffusion, and visual latex agglutination. This is the basis of the CRP test. We have an latex that antibody against CRP is coated on its surfaces, and if we add our sample and the CRP is present in our serum, it leads to the agglutination, and this agglutination can be seen by eyes. Today we want to use the CRP latex test kit. This kit contains CRP latex reagent, CRP positive control, CRP negative control, and one of the circles you should use as test slide. You add the different solutions to the slide, and based on the level of agglutination, you can denote them, the plus scores. After adding the different controls or serum to the slide, you should rotate in these rotations on the desk to doing better this test. The next and the last experiment today we want to talk about them is rheumatoid factor. Rheumatoid arthritis is an autoimmune disorder that occurs when the body's immune system mistakenly attacks its own body tissues. This is one of the type 3 hypersensitivity in our bodies. High level of rheumatoid factor occur in rheumatoid arthritis, and rheumatoid factor is an antibody that produced in rheumatoid arthritis patients. It is defined as an antibody in class IgM and can bind to the Fc region of class IgG of antibody. RF and IgG join to each other and make a complex that this complex can deposit in our joints and other regions and ultimately leads to the disease. As you see in this slide, we have an antibody from class IgG. This is the Fc region, and this is the Fab region that can bind to the antigen. In the rheumatoid arthritis patients, antibody produced from class IgM can bind to the Fc region of IgG, and this complex can deposit in different region of body and leads to the disease. The basis of the test is very similar to previous test that we say in CRP. Antibody is coated on the surface of the latex. Then we add the serum of patient. If the patient's serum have pentamer IgM that can bind to the Fc region of IgG, it's lead to the agglutination in our slides. This kit is similar to the CRP test and have human IgG latex antigen, positive control, negative control, and test control, and the others is very similar to CRP test, and you should, after adding the different solutions, you should rotate the slides to combine the different agents in the slides. Thank you for your attention. If you have any question, I'm in the lab and I can direct you. ASO buffer. Each two droplets is 0.1 milliliter. Okay. For the tube 1, we should add 0.2. 1, 2, 3, 4. For tube 2, we should add 0.4. Tube 3. Okay. 6. Tube 4. 0.7. Okay. Tube 5, 1 ml. Okay. And finally, tube 6. Okay. Then we should add the serum. For tube 1, 0.8. Okay. Tube 2, 0.6. Tube 3, 0.4. And for tube 4, we should add 0.3. Okay. For tube 5, as our control negative, and tube 6 as our control positive, we don't add any serum. Okay? Then we add RBC in the same amount for all tubes, 0.5.
पता है ना कितने ड्रॉप्स नहीं बट इस पे फिर दिया ट्यूब्स पे लिखा क्या सब वाला
Okay. We have 5 minutes of incubation, and then you should add the same amount of Other thing, yeah, of course.
RBC.
If we have a problem, we should don't add RBC, we should add the SLO to this, you know, and after 5 minute incubation, you should add the RBC. So you do the correct thing. I do a mistake here, and after adding serum, you should add SO. You know, same amount to all of tubes, based on the procedure. Then 5 minute incubation, add the patient serum, okay, serum, and here serum, okay. Then you add the buffers for CRP in place 1 and for RF in place 2, okay. Then you should report the result for us, okay. If you see the agglutination like this, okay, this is the result of 1. Agglutination like this, you should report the positive. This patient serum is positive for CRP. And also for RF, the result is like the number 1, okay? If you say the agglutinations and the complex, you should report as positive result, and if you don't see any complexes and depositions in your slide, you should report as negative serum. This is very simple experiment. Okay? Then you add 0.1 of serum to tube number 1. Okay? Then you should, after adding the serum to tube 1, you should transfer 0.5 from this solution, this combination, to tube 2. Okay? Then from tube 2, you should transfer 0.5 to 3, from 3 to 4, from 4 to 5, and from 5 to 6. Okay? Then you should add 0.5 from antigen to each of them. It is the same for all. Yeah.
15.
0.5, all of them is 0.5. Okay, this is okay? Okay. The last tube, you see the agglutination is the dilution you should report for us. Okay, the last tube you see agglutination. You can see the complex that formed by your eyes. The last tube, for example, if the last tube you see the agglutination in the solution, you should report 106. Okay, and the dilution more than 160, 1 to 160 is related to the patient serum. Okay? Okay, you can initiate, give your reacts from adding 0.5 from tube number 5. You should spill over 0.5 from tube 6 to all of them being 0.5. Okay? I think. Okay. And to all of them, to all of the 2, 3, 4, and 5, you should add 0.5. 10 droplets.
He's adding 0.5.
Okay. Then we add 2 droplets of serum to tube 1. Okay. 1, 2.
Okay, and then the next ones are.
Okay. We transfer the pipette. Okay. Mix them, and then transfer 10 droplets to tube 2. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then
Return
we
it.
should do this
Remove.
from tube 2 to tube 3. We mix first,
It's just
then
how
transfer.
Hamid explained.
How many?
10 droplets. Okay.
Could you hold it, please?
Just check it's okay.
Yes.
Okay.
Now I can see.
Okay. So the last tube in our experiment is tube 5. So we split over 0.5 from it to 5. 1, 2, 3. Okay. Then you add antigen in same amount.
How many?
0.5. Okay. Then after you should give this to Mrs. Moradi and change the tubes for speed of the experiment. You have incubation in this period, so to refuse the incubation, we process them from yesterday.